Great. Welcome everyone to our CFAR seminar this month. We've got a great lineup today. An early stage investigator presentation by Jesse Deer, followed by a plenary talk by our invited speaker, Ann Sharuti. We're really delighted to have with us today. Let me start by introducing Jesse. Jesse is a project scientist in the Department of Medical Microbiology and Immunology in the School of Medicine at UC Davis. And he works with Dennis Hartigan O'Connor's group. His research focuses on the mechanisms of viral persistence, in particular the mechanisms utilized by HIV and CMV. And the overarching goal of his research is to develop better prevention and intervention strategies to disrupt virus host relationships in favor of the host and viral clearance. And he's especially interested in HIV persistence during ART, including viral reservoirs and cure strategies, which makes him a great pairing with Ann's talk today. He is also interested in the many ways that CMV manipulates the host to establish lifelong infection. So with that, I will let Jesse get started. Thanks again for joining us, and take it away.
Okay, so I'm Jesse Deer, and I'm a project scientist in Dennis Hartigan O'Connor's lab. And today I'm going to talk about some of our recent work in the neonatal macaque model. So the title of our talk is SIV clearance from neonatal macaques following transient CCR5 depletion. Okay, so the focus of our work is, let me see, make sure I'm going right way. Okay, so the goal of our work was really to try to determine whether debulking the HIV reservoir is a viable path towards achieving HIV remission. And really there are two different strategies to do this. One is through latency reversal, where we use LRAs to disrupt the latent reservoir and then rely upon the host immunity to kill the infected cells as they express their genome. And then the second approach, which is what we wanted to work on, was a direct depletion strategy. And so this is a less focused approach. And the goal is really to target surface markers on target cells to wipe them all out. And so other people have sort of talked about this idea, and reservoir reduction strategy was presented as a model by Hill and Silicano and colleagues. And they showed that, get my laser pointer going here. And their model showed that there was an estimated 2,000-fold reduction would be required to permit a median 1-year ART-free remission. And so that's in the gray line here. And that a greater than 10,000-fold reduction would prevent rebound altogether. And so this work provided benchmarks for us for viral reduction to achieve HIV remission. And it also warned us that the bar was pretty high, in terms of the degree of depletion that would be required to have an impact. And so the hypothesis of our work was in early SIV infection, transient elimination of CCR5 positive cells that are infected or susceptible to new infection is safe and can delay rebound after analytical treatment interruption. And so then the goal was really to determine whether direct depletion could debulk the reservoir and impact viral rebound. And so this included the two different mechanisms. One would be the elimination of infected cells, and then the second would be to eliminate targets for infection. And so we were interested in testing two different agents in this project. And the first is an anti-CD4 antibody, and then the second is an anti-CCR5 CD3 bispecific antibody. And that's what's shown in the cartoon here, where the antibody recruits CD8 cells to target cells to initiate killing of the target cell. And so our experimental design, we inoculated macaques, neonatal macaques, that were about between 2 and 3 weeks old, with SIV MAC 251, and they received 2 challenges, one in the morning and one in the evening. And then 7 days later, we initiated ART. And then 2 days after the initiation of ART, we depleted them with either our CD4 antibody or our bispecific antibody. And we kept the animals on ART for 18 weeks to allow for viral load suppression, and then removed ART to look for viral rebound. And so in terms of depletion, we assessed cellular depletion following antibody administration and showed that our depletion was complete. So for the CD4R1 group, they're in blue. The bispecific antibody group is in red, and we're showing absolute T cell counts on the y-axis and time after antibody administration on the x-axis. And so one of the things we were really surprised about was the durability of the depletion that we achieved with our bispecific antibody, with some animals not reconstituting until 9 months after the single antibody administration. And with the CD4R1 antibody, we had achieved a comparable amount of depletion, but it wasn't quite as durable, and they tended to recover and reconstitute their cells more quickly. But I think it's important to point out that the depletion was transient, and that both CD4s and CD8s were restored eventually, and that we didn't observe any adverse health effects in the monkeys. And so next we measured plasma viral RNA to determine whether we were impacting that with our depletion. The shaded box indicates the time on ART, which was 18 weeks with our 3 treatment groups. And the first thing we noticed was that with the bispecific antibody, we saw a steeper decline of viral RNA following target cell depletion, particularly steeper than the ART alone group. But it was also steeper than the CD4 group, which actually looked like there was some depletion too. And then following the removal of ART, we saw a significant delay in rebound in 4 out of 7 of the bispecific antibody treated groups. And so the ART alone group, those all resoundingly rebounded within 2 weeks of ART withdrawal. And then the same was true for the CD4 group, except for a single animal that didn't rebound after removal of ART. And so this was a surprising result for us. We didn't expect this degree of delay in rebound almost universally across the group. And then as we followed these monkeys over time, we had 2 monkeys that failed to rebound at all. And one of those we sacrificed earlier in the study to assess the viral reservoir. And then the other one we continued for 3.5 years. And we've just recently sacrificed this monkey too with a full necropsy. And we're currently working on going through the tissue reservoir to determine whether we can find any evidence of infection remaining. But before we sacrificed them, of course, we treated them with CD8 and anti-CD8 antibody to deplete CD8 T cells, and that didn't seem to have an impact on the remission in either of the monkeys. So we were interested in whether the delay in rebound that we observed was significant or not. And so we did a Cox regression analysis that was corrected for the peak viral load to account for any differences between the groups there, and found that the bispecific antibody treatment was statistically significant. The difference was statistically significant in time to rebound. So next, I'm just going to kind of hustle through a few slides that show some of the reservoir analysis that we've done to date. And I don't think I have enough time to really delve into it, but I just wanted to highlight a few key things. And so we've done qPCR from tissues, from multiple tissues for both viral RNA and viral DNA, from the no rebound animals that we sacrificed and did a necropsy on. And we failed to find either viral RNA or DNA in those animals. We've also done RNA and DNA scope on tissues from them. And despite finding it in our control animals, we did not find it in any of our aviremic animals that we assessed. And we've since this slide, this work was done by David Merriam a graduate student in our lab who's moved on. And since the necropsy on our most recent animal, we've sent tissues up to Jake Estes at OHSU, when hopefully we'll have a more in-depth RNA DNA scope analysis from these tissues and the newer animal too. And we also did viral outgrowth assays on lymph node cells that we collected at lymph node biopsies longitudinally, and then also from large numbers of mesenteric lymph node cells that we purified at necropsy. And those viral outgrowth assays have been negative too, despite being positive in our control animals. And so for conclusions, I just want to say that our anti-CD3 CCR5 bispecific antibody provided delayed rebound as well as long-term remission in 2 of 7 of the treated animals started on ART at 7 days post-infection. And the remission was achieved in 2 different experiments in consecutive years. And so we feel like we've successfully shown reservoir depletion debulking, and we had clearly delayed rebound kinetics in all of the bispecific antibody recipients. And so we think that this demonstrates the potential importance of target cell depletion as an HIV cure approach. And one of the things we were wondering, because we didn't achieve a 10,000-fold reduction in target cells, it was more like down to 1 cell. And so we're wondering whether the depletion of uninfected susceptible target cells contributed to our effect, which wasn't accounted for in the model by Hill and Silicano and colleagues. And so we feel like we've demonstrated that CCR5 depletion was safe and well tolerated, and it was surprisingly durable but transient, and that any unanticipated risk of CCR5 depletion presumably would also be transient. So we think that we've shown a single shot cure in 2 senses. 1, administration of the drug and no requirement for any additional latency or reactivating agent. And so next steps and future directions, will the strategy work if the reservoir is more established? Unknown but quantitative differences in the reservoir might be addressed by administering more than 1 dose of drug. And we also have postulated that we could find greater potency of alternative drug designs. This was just our first approach. And so we're also wondering whether CCR5 depletion could prove synergistic with other modalities, such as therapeutic vaccination or latency reactivation. So I just wanted to acknowledge and thank everybody that's contributed to this work. These cure projects take years, and it's a lot of effort, and almost everyone in Dennis's lab has contributed in some form. And we have collaborators across the country that have done really important work and are doing that for us now. And with that, I just want to say thank you for your attention.
Wow. Thanks very much, Jesse, for a really amazing presentation. We have maybe just time for 1 or 2 very brief questions. I may just jump in with whether you think there might be an effect on an X4 using a viral variant, or is this only going to be effective, you think, in people who harbor CCR5 tropic variants?
Yeah, that's a great question. And we haven't addressed that specifically yet. And, these monkeys were treated really early in infection, at 7 days. And we have another monkey that's currently on study that we've allowed to go much longer before we initiated ART. And we haven't really assessed the tropism of that virus, which would be interesting to do. But yeah, that's a good question.
Yeah. And then Philip has a question.
Yeah, I do. Thanks. So there's some variability in how long the depletion lasted, and then also variability in the time to rebound. And did that correlate? So if a monkey had a longer period of depletion of the target cells, did that correlate with those who took longer to bounce back?
We haven't looked at that specific correlation. Well, actually, Dennis is telling me that it did not correlate. Did not correlate.
Okay.
Yeah.
Okay, great. Well, great work. That's a great talk. Thank you.
Thanks.
All right. Steve, maybe you want to get something in real quick?
Jesse, there's a lot of virus in CCR5 negative CD4 positive T cells. And so what happened to that virus? What happened to those reservoir cells? Were they depleted too? Or were they left behind, and why didn't they rebound?
Yeah, it's a good question. One of the things we had considered is that there could be a blocking effect from our bispecific antibody, just occupying the R5 receptor on susceptible cells. So if it didn't necessarily deplete them, it could block them. And so maybe that contributed to some of the reduction we saw, virus coming out of the other reservoir cells, maybe didn't have a target cell to infect and
Yeah.
succumb to the immune system or, phased out. I mean, that's a great question, though.
Okay, great. A really terrific presentation, Jesse. And maybe you want to shift over into introducing our next speaker.
Yes. 1 second here. Okay, so it's my pleasure to introduce today's speaker, Dr. Ann Cerruti, who is a leader in our field. And she's a tenured professor of pediatrics in the Division of Pediatric Infectious Diseases at Emory University School of Medicine. Dr. Cerruti directs the Center for Childhood Infections and Vaccines of Children's Healthcare of Atlanta and Emory University, and is the co-director of the Basic Science for Emory Center for AIDS Research. She's also the founding director of the Pediatric Residency Investigative Scholars at Emory program. Dr. Cerruti earned her MD and PhD from Emory University School of Medicine and completed her general pediatrics residency in the Children's Hospital of Philadelphia. She returned to Emory for subspecialty fellowship training in pediatric infectious diseases. Dr. Cerruti's NIH-funded research program focuses on HIV persistence and remission using non-human primate models to evaluate translational interventions towards an HIV cure. She received an HIV NIH Merit Award in 2021 for this work and is MPI of the Pave Martin Delaney Collaboratory.
Thank you so much, Jesse. You can hear me okay? All
Yep.
right, great. Thanks, everybody. It's great to see so many familiar faces, and that was really a super great presentation and such a wonderful example of how infant non-human primates and non-human primates in general are can be such a valuable resource in testing these exciting new strategies to try to reduce reservoirs and lead to post-treatment control. So I'm going to kind of expand on that a bit and talk to you about some of our work in pediatric populations towards an HIV cure. And I tried to, with this talk, sort of put some of the monkey work and also some of the related studies in children living with HIV or infants living with HIV to try to give both perspectives. So let's see. Okay, I've got a couple of disclosures there. And I thought actually I would just start with a case just to kind of bring it home to some of the more clinical scientists here and just to kind of tell you, I have two cases kind of interspersed throughout this and they give you, I think, a good picture of the types of individuals that we're seeing here in Georgia in the current and ongoing HIV pandemic. So this is a 6-month-old female from rural Georgia. Her mom received prenatal care. She was informed that she had tested positive for HIV at 7 months gestation, but for some reason that we have not figured out, she was not started on any antiretroviral therapy until after the baby was delivered. She did get zidovudine intravenously during her labor, and the baby was born by C-section at 38 weeks and 2 days. So then the pediatrician who saw the child started the baby on triple antiretroviral therapies, zidovudine, lamivudine, and nevirapine for prophylaxis. And these are all sort of prophylaxis / treatment because it's treatment doses that we use. And the baby tested negative at 15 days and 6 weeks of life, but then positive by HIV DNA PCR at 5 months of life. And a confirmatory test was sent and viral load came back at 6.7 million copies with a CD4 count quite low for an infant, 256. In addition, kind of over the course of this time, the baby had recurrent otitis media and gross motor delay. And then kind of once all this came to be and that the diagnosis was confirmed, then we restarted antiretrovirals as well as Bactrim, and the child is doing relatively well now, no new infections or concerns, and the viral load is trending down slowly. And I just put this case up to demonstrate that unfortunately HIV, perinatal HIV transmission is still happening here in the United States, while of course the vast majority of those transmissions are happening in sub-Saharan Africa. We still do have to always be vigilant about our preventative measures. And so a little bit more, just to kind of start off with some background on perinatal HIV and how it might differ from adults who acquire HIV through sexual contact or intravenous drug use. What's important to kind of recognize is that, oops, sorry, that's not what I meant to do. Laser pointer. All right, I'm just going to use my mouse. What's important to recognize is that, you know, infants who acquire HIV perinatally go through many different stages developmentally and for their immune system in terms of the development, and things change over the course of that period of time, and behaviors change as well. But this is kind of a very brief summary of what's happening in terms of the immune system over that period of time. So early in life, the immune system is more prone to be tolerogenic, skewed towards regulatory CD4 T cells and Th2 versus Th1 skewing as well. There's a reduced and kind of a less polyfunctional and lower breadth antiviral immune responses or adaptive immune responses that has been demonstrated in perinatal HIV. Matthias Lichterfeld and others have highlighted the importance of the innate immune response to HIV control in this period of time. And then I think what's really sort of interesting, but not really very well studied, is the influence of sex hormones in puberty. And one other thing I just wanted to point out is that, you know, something that could be considered a great advantage towards a cure in perinatal HIV is that we do treat very early. We treat them as soon as they're born, basically. And so it's feasible, and that does result in low reservoir size, limited viral diversity, but that also impacts the development of HIV-specific immunity. So you have less antigen around that can then lead to these reduced adaptive immune responses. Okay, and we all know this, I think, on the call, but I'm just putting it up there to remind us of the anatomic sites that HIV persistence has been demonstrated to exist in, and the different cell types, as well as the mechanisms. And this is a really nice figure that's from, you recognize the first author of this paper here. And then on the right side of the screen is sort of some kind of a nice Venn diagram of where the reservoir has been demonstrated to persist in terms of different types of CD4 T cells, and related to their memory state, and related to their helper polarization, excuse me. And then one thing I wanted to, the reason I have this up here is because you see these naive cells over here. And this is something that we have been interested in, and others have been interested in for quite some time, is the role of naive CD4 T cells in perinatal HIV infection, because you know, you may know that the CD4 T cell compartment in infants and children is the vast majority of it is comprised of naive CD4 T cells, and that is shown here, kind of across age in both on this slide, on this picture on the left, infants to seniors, here infants to adults, and then looking at different anatomic sites and the composition of naive versus memory and other CD4 T cell types. So what you can see is in the blood and lymph nodes, as well as spleen, there are really sort of in infancy, naive cells are the vast majority of CD4 T cells, less so in the intestines and the lungs. And this is kind of showing a similar picture, but also looking at their antiviral or antimicrobe specificity. So the question we had asked is, what is the potential contribution of naive CD4 T cells to the HIV reservoir? And we looked at this in our non-human primates. So this is a study we did now several years ago, where we looked at both infants and adults that had been infected with SIV and treated with antiretroviral therapy. And in this case, it was about 6 months of treatment. Then the animals were euthanized, and both peripheral blood and lymph nodes, although I'm not showing lymph nodes here, were isolated, and CD4 T cells were sorted into different subsets. So naive stem cell memory, combo central transitional memory phenotype, and effector memory. And similar here in the adults, although the central and the transitional are separated. And we just looked at the level of total SIV DNA. So this is both, intact and defective, although in the non-human primate model, the levels of intact and defective SIV DNA are much more similar than they are in humans that have been treated with antiretroviral therapy for a long time. And basically what we found in this kind of small cohort study is that there were similar levels of infected cells across the different subsets. In the lymph nodes, we did have a slightly higher level of infection in T follicular helper cells compared to the other subsets. And in the adults, we saw that while we could find virus in naive CD4 T cells, it was at a lower level than in all the other subsets. And so when you do the calculations of looking at the contribution of each subset to the pool and then take into account the level of infected cells, you can sort of see here that the naives are comprised about 3/4 of the reservoir in the infant peripheral blood compared to a much smaller proportion in the adults. And this is just a very small 2-animal QVOA where we looked at the ability of naive CD4 T cells to produce replication competent virus that could go on to infect new cells in co-culture. And so we did indeed find that. So, and I won't go into this in great detail because it's very similar. This is looking at infant macaques that are infected with SHIV virus. This is SHIV CH505. And so we did a very similar analysis looking at the levels of virus across different subsets and then looking at their contributions to the CD4 T cell pool in blood and lymph nodes, and then their contributions to the CD4 T cell reservoir, and had similar results than looking at intact in naive versus memory cells. So naive, the kind of conclusion that we drew was that in these infant macaques, the naive CD4 T cells can be infected. It's at a lower rate than the memory CD4 T cells when you look at the intact SIV or SHIV provirus, but they can produce replication. They can contain replication competent virus, and so they should be something that we consider both. We're very interested in how they're getting infected, but then also do we need to be considering them when we're thinking about cure strategies? And for example, these cells do not express CCR5. So this would be kind of interesting to see, what happens with them in Jesse's study that he presented earlier. Okay, so then now I'm moving on to kind of a corollary with a human study that we did recently. And so this is a study where we wanted to look at the naive infection in children who are living with HIV. So this is showing our different study participants and the subtype they're infected with, how old they were at ART initiation, which was a median of 9 months of age, and how old they were when we collected their blood to do this analysis. So they were all 10 and younger and a median of 8 years old. These are we collected their blood and sorted naive and memory cells from their PBMCs. And these are the markers that we use to sort. So we tried to be very strict in our assignment of naive versus memory, and tried to be very conservative in terms of setting gates to not have any contaminating memory cells in the naive population, because that's often a concern with these types of studies. And so our purities are shown here, and I'm not going to go into it, but a collaborator, we did all of this work in collaboration with Mary Kearney's lab at the NCI, and one of her biostatisticians that she works with, Rayson Rausch, did a tried to look at the purity of our sorts and the potential for contamination that when I show you the kind of level of infection and whether that could be related to contaminating memory cells, and the overall conclusion was that that is very unlikely to be the primary dictator of the infection data that I'm showing you next. Okay, so initially we just looked at, or Mary's team just looked at the level of infected cells by HIV LTR PCR. And so the overall infection was 38 per million naive CD4 T cells and 464 per million memory CD4 T cells. So about 12-fold lower in the naives, which is not unexpected. And then when she looked at looking at the LTR, Psi, as well as RRE, so these are the presumed intact proviruses, so the sort of an IPDA, they were able to detect that 4% of these 201 had all 3 of these targets were able to be detected. So we have this 8 out of 4.5 million that were presumed to be intact within this is just showing the naive cells. They went on to do some integration site analysis, and there's a lot more of this than I'm going to show today, just in the interest of time. And also there's going to be a presentation from her group at CROI about this. So I'm kind of saving some of that data for that time. But they did sort of a deeper dive in this one particular individual and looked at the integration sites in both the naive and memory CD4 T cells. And I'm just showing you the naive here. So they were able to find 85 unique integration sites within those naive CD4 T cells, and 61 of them were unique. They did find that 24 were detected more than once, and they were able to identify 8 different distinct clonal populations that are all shown here. And this one was the largest clone was 7% of the infected naive cells. And so one of the things that we were, she's very interested, we're sort of comparing the integration sites between the naive and the memory cells and looking at the relationships between the 2 of them. And I won't give any spoilers, but I think we're finding some very interesting things from those data. So just to kind of summarize this portion, we are showing you that there are infected naive CD4 T cells in both ART-treated SIV and HIV infections. We can see intact and replication competent virus in these naive CD4 cells, as well as clonal populations with integrated HIV were detected through the work with Mary Kearney's group. And so one of the things that this leads us to think about and question is sort of, because we have such a high population of naive CD4 T cells in early infancy, as well as that regulatory or tolerogenic immune environment, you know, that those two combined likely influence the composition of the reservoir, but potentially also the reactivation potential of the reservoir. And I'll kind of talk you through a couple of slides on how we are looking at that reactivation potential. But I just wanted to, there's a bunch of references down below here where we're not the only people who have identified naive CD4 T cells. The lab of Una O'Doherty has been studying this for some time in terms of their contribution to the reservoir in adults, but there's also a number of other papers going back many years, and some papers that didn't necessarily highlight that the naive cells were there as infected or a reservoir, but when you look at the data as compared to the memory cells, they definitely you know, have shown either intact or just kind of the presence of viral DNA. Okay, so in terms of the reactivation potential and how that might be how that might be different in infants versus adults, I'm just briefly introducing one of the latency reversing agents that we've studied, AZD5582, which is a SMAC mimetic or an IAP inhibitor that activates the non-canonical NF-kappa B pathway shown here. And the eventual result is that you have an active dimer that's in the nucleus that can interact with the HIV LTR and lead to transcription and viral RNA production. And we've looked at this both in infant and adult monkeys, and probably this is our study over here where we looked at it in the adult monkeys, and you may be familiar with this figure, but this is from the first study that we published, and this is showing you the levels of latency reversal or virus reactivation, or on-art viremia is another way that we've referred to it before. But these are animals that had been suppressed on antiretroviral therapy for over a year. They are treated with this SMAC mimetic once a week, and then we're also still giving them daily antiretroviral therapy. And then we're seeing that they have, you know, previously they had been, I'm not showing you that, but previously they had been suppressed for a long time. And now we're seeing that they're having these viral RNA in the plasma. Now, on the left, when we do basically the same study in infant rhesus macaques, we see we do see some viral blips here or on-art viremia, but it's much lower than what we see in the adults. Less well, I wouldn't say lower in terms of the level that we can achieve, but in terms of the frequency of events of on-art viremia, you can see by eye, it's quite different. And so one of the potential hypotheses as to why that might be is that this agent differently impacts CD4 T cells in infants versus adults, or perhaps that the naive CD4 T cell reservoir contribution is less sensitive to the AZD5582. And I don't have a definitive answer on that yet, but we are exploring that further. We did look at activation of not the non-canonical NF-kappa B pathway, or in general, we looked at sort of the response of CD4 T cells to these to the AZD5582. So we isolated CD4 T cells from the blood of these animals before and after the 10-dose treatment. And then we looked at the gene expression changes. And the way that these heat maps are organized is that this list of genes here were similarly either up or down regulated in the infants and the adults treated with AZD5582. This list here in the middle shows you the genes that had higher expression higher fold change expression in the infants compared to the adults. And then on the right, the converse, so the higher expression in adults compared to infants. And we noticed that a couple of the non-canonical NF-kappa B genes, NFKB2 and REL-B, were they still were upregulated a bit in the infants, but it was much stronger in the adults. And so that's potentially what's driving the reduction in latency reversal that we're seeing. On the one hand, we do have some data that perhaps the metabolism of AZD5582 is different in infants, and that we actually need to increase the dose to reach a similar Cmax. And so that may be the pure example or the pure explanation for why we have these differences in latency reversal. But and we're actually, I'll show you in a few slides, we're embarking on a new study at a higher dose, and we'll be able to see if that is really the main driver, or whether we can learn anything about the relationship of naive versus memory cells and their responsiveness to the SMAC mimetic or even other latency reversing agents. Okay, so in the last bit of time, I'll kind of dive into some interventional trials. And this is a table that I stole from Debbie Prasad. I apologize, I didn't put her name down here, but it was made in 2022, but it's really not any different today. These are the clinical trials that are studying either very sort of cure interventions in quotes in children. The first 4 are all just very early ART, nothing else. The next 3 are using early ART with BNAbs, and then you're familiar with the transplant study, although there haven't been any children that have been enrolled in that to my knowledge, but that I might be wrong about that. And then there's this therapeutic vaccine study that's getting underway in Europe, South Africa, and Thailand. So because we've been doing some work looking at antibodies and their role in HIV, post-treatment control of HIV or post-ART control and reservoirs, I just wanted to show you one slide on the Totelo study. And I guess the point here of this, before I move on, is just that this number 9 is pitiful. It's very small, and we, Dennis's group, my group, and others are hoping that the work that we're doing in the infant monkey models can really add to the number of studies we're able to do safely in infants and children. Okay, so the Totelo study was recently published this past year, and this was a study of dual HIV BNAbs in children. They had been previously enrolled in an early infant treatment study in Botswana. They had been on antiretroviral therapy, and I think they were around median of 3 years old at the time that they then shifted over into Totelo. And during this time, they received a period of antiretroviral therapy plus broadly neutralizing antibodies, VRC01 and 101074. They were given initially with ART and then once a month with during ART interruption. And so then the main goal of the study was to look at how long viremia could be suppressed. And so I'm not going into all the details about kind of what determined who was successful or not, but suffice it to say it was just under 50% that were able to stay off of ART for 6 months with monthly BNAb treatment. So I think it's not surprising that happened, but this was, sort of the first dual BNAbs trial in children. And so it was important. And I think we are learning more, and they're pushing the envelope in terms of adding on to this study and potentially looking at additional antibody therapies. So in my lab, we had been kind of simultaneously looking at antibodies to give to animals that had been treated with antiretroviral therapy and see kind of what their impact on rebound would be. And so this was a study that we gave SIV envelope specific antibodies that had been derived from rhesus macaques from the group of Mario Roeder and Rosie Mason. And we gave it a week. We gave this combination a week before treatment interruption, and they basically just followed the animals to see the time of rebound and the rebound kinetics and dynamics. And so what we were able to show is that in the animals that received the antibodies, they had a delay in their rebound. So again, not super shocking or surprising. We only gave 1 dose of the antibodies. So, because we just wanted to sort of get a baseline of how long it would be until rebound. And I'm showing you here the percent that had rebounded. So we had a delay of about 45 days. So that is in keeping with potentially giving antibodies on a monthly basis, as was done in Totelo. Now we're learning more about kind of what were the predictors of rebound and how they relate to the antibody targets. So we had 2 antibodies, that ITS 102 and 103, that target the CD4 binding site. This is an Emperor specific antibody, and this is a V2 specific antibody. These are their concentrations in the infants over time, and you can see it's kind of variable. And this antibody in particular is we've shown in adult monkeys that it sort of induces ADA most rapidly. And so I think we haven't shown this yet here, but I think that's the rationale, or that's the reason why the level concentrations drop off much faster than the others. And so this basically what we can conclude from this, and we've done some correlative studies that sort of correlate the antibody concentrations to the time to rebound. And it appears that the V2 and the Emperor specific antibodies, somewhat surprisingly, are actually the ones that are most predictive. Their presence is most predictive of kind of maintaining viral suppression off of ART. And so this is something that I think was an interesting study to do. I think was important for us to kind of learn more about how these antibodies affect rebound. But in the end, what we want to try to do is to use these interventions to clear infected cells, to reduce the size of the reservoir, to do, what Jesse and Dennis are working on as well. And so, we know that bNAbs can neutralize virions, but they also can participate in clearance through ADCC or ADCP, complement-dependent cytotoxicity, and this potential bNAb vaccinal effect. So I'm going to show you, some data from a study that is in process, where we're using that antibody approach as a clearance approach, and combining it with the SMAC mimetic AZD5582. So this is the classic shock and kill, but as antibodies as the kill mediator. And the particular antibody that we're using in this initial study is ECD4. So, the details of ECD4 are really shown here. It's very appealing for us because it works against both HIV and SIV. It does not, or it recognizes epitopes on both HIV and SIV envelope. It's very challenging to develop resistance to it. It has an FC domain that we can utilize for interactions with NK cells for the ADCC, et cetera. And so this was kind of our first trial that we wanted to test out as with the shock of AZD5582. And delivery. So the delivery method that we're doing is adeno-associated virus. So we didn't want to have to do repeated injections because we are worried about developing ADA and sort of, that the need, thinking ahead, the need to have monthly access. And so we used AAV vectors that will deliver the ECD4 to these infant non-human primates. I'm going to skip over this in the interests of time. Okay, so here is the study design. We have animals that are infected with SIV, and then at 3 weeks after infection, they're initiated on antiretroviral therapy. And in the experimental group, they get the AAV9 that is encoding ECD4 IgG1. They remain on ART, and then they will receive the SMAC mimetic AZD5582. So the idea is that when we see latency reversal here, the ECD4 IgG1 that's circulating in the animals will then recognize those reactivated cells and hopefully eliminate them, and we'll see a reduction in the reservoir size. I'll say we're kind of like right at this stage, so I don't have data from here, but I just wanted to show you expression data to hopefully pique your interest about where this study is going. And we have, like I mentioned before, done some additional PK studies and some modeling to make sure that we're actually giving the animals a dose that is going to hit the adult Cmax. Okay, and these animals were screened for pre-existing neutralizing antibodies to AAV9 so that we could kind of set ourselves up for success. And here's the data that we have so far. In red, we have the viral loads of the infants. And then in green, we have the ECD4 IgG1 expression. And so we're pretty excited about these levels. In most animals, they've come up and stayed up. Some of the animals are on different time frames. So I don't have, like for this one started much later. So we're really at the point for the earliest ones that we are going to be giving the AZD very soon. So those reservoir data are forthcoming. All right, so I tried to take you through a lot of stuff in a short amount of time. So hopefully there was that piqued your interest in some of the work that we're doing. So I talked about the persistent reservoir in both naive and memory CD4 T cells, and the work from Mary's lab looking at intact integrated clonal HIV in children with perinatal infection. This idea of reduced latency reversal in infants compared to adults, and maybe related to drug metabolism purely, but we're also really interested in exploring more about how the naive cells may be contributing to that as well. We know we've shown, and many other studies now have shown that bNAbs delay viral rebound, but they have to be given repeatedly. The concept that AAV gene therapy holds promise for inducing persistent expression of bNAbs. I'm going to skip over this clinical vignette. I included it because it's another example of an adolescent that I follow in my clinic. And well, actually, I kind of want to show it. So I'm just going to take time, 1 minute to do it. And then hopefully there will be still some time for questions. But this is somebody who benefited from the work of the UCSF CFAR because he was an individual who had perinatal HIV, previously had been very well controlled, but really struggled with adherence for a number of years, and did not have extensive resistance. And due to some of the work that Monica has presented on injectable cabotegravir rilpivirine in individuals who are viremic, we applied to his insurance company to get approval to give it to him. He went from 100,000 to 100 after a dose, and then he's been suppressed after 5 doses, and he's remained so. And so that's just super exciting for him. And I put him in here because he's sort of the other spectrum of pediatric HIV and perinatal HIV. And most of our attention in the pediatric world is focused on the infants. And I'm hoping that your attention in the adult world can be focused on cure in these guys. I think that they are not excluded from adult HIV cure trials, but I'm not sure how much they're included either. So I just kind of wanted to hear what you guys thought about that. And with that, I will thank everybody who's on this slide, collaborators, my lab, and our funding sources. And thank you all for this time.
Wow, Anne, that was terrific. Thanks so much. While people are getting ready to raise their hands, I can start. Your story about decreased latency reversal in naive cells really resonated with me. Nicolas Chaumont had some data with his TILDA assay years ago showing that effector cells, you can more readily reactivate virus from effector cells than less differentiated cells. Matthias Lichterfeld, I think, had a similar story
Mhm.
about T cell differentiation. And then in this last year, we've shown in our HOPE study that viral reservoirs actually decay more quickly in effector T cells than they do in less mature T cells during antiretroviral therapy. So sort of very consistent with what you're seeing there. And this is probably more of an issue with the kids than it is with
Yeah,
adults.
yeah. No, and we are sort of have a project now where we're trying to look at the sort of really, look at isolated naives versus memory and test different latency reversing agents and really sort of, assess whether that hypothesis is true. That's kind of seems like it's being, there's a lot of different experimental data that's pointing to it.
Yeah, great. Steve Uckel has his hand raised.
Yeah, can you hear me?
Yes.
Sorry, my computer's turned. I actually was going to make a lot of the same comments Peter made, but another thing is in our studies that we did in the HOPE trial with Peter, we looked at the baseline HIV transcription in different subsets of CD4 T cells. And I can't remember all the data, but my recollection is that the naive cells were transcribing less HIV RNA than the other cell types. And I think it was because they were initiating less HIV transcription. So that may also be, they may have differences in the factors that initiate HIV transcription, and that may partially explain the lower response to that AZD drug or other IAP inhibitors. Just a thought, not sure. You'd have to test it, but.
Yeah, no, thank you. That's really interesting.
Others, Monica has her physical hand raised.
No, I'm so sorry. They don't let me raise my hand because I'm a host or something, and I don't know. But that was great, and that was so interesting. I have a question actually for all of you who work on cure and latency in the context of long acting. Do you think that long acting drugs, because their PK is such that it just keeps on going down with time, there's not high competition of the PK of a Cmax every day to fight the virus and to get it to kill it every day? Do you know what I mean? Like it just keeps on going down. Do you think that there'll be more inflammation and a higher reservoir with long acting antiretroviral agents than a daily pill that goes up and down every day? This is just theoretically something I've been concerned about. I know it's a theoretical question.
I mean, I maybe Peter, you're probably better able to answer that than me, but I think at least my understanding of the pharmacology, which is minimal, is probably that the idea of the long acting is that you hopefully achieve, a high enough level. Obviously, it's keeping the virus suppressed that might not be a problem. But I wonder, have people looked at residual viremia in those individuals? I don't know.
This is just emerging because we don't have enough demonstration projects and using it in viremic patients. But in the OPERA cohort, when they used long-acting ART, 82% got to less than 50 copies per mil, but 94% of 176 people got to less than 200 copies per mil. And I just wonder if it's allowing for residual viral replication, especially near the end of the dosing interval. I'm just very curious about PK and inflammation and viral dynamics with long acting, which we're just at the tip of thinking about it. And like you showed in your last case, long acting can be very important for adolescents.
Yeah. Oh, it's a really good point.
It's a question to put out there. Yeah. Thank you. That was a great talk.
Steve.
Or Monica, I would guess the peak, the way that the way that these long actings are dosed is to maintain levels above, whatever is needed to fully suppress the virus. So I would doubt you'd see a lot of blipping or residual viremia with ARP, but BNAbs, long-acting BNAbs, we're using it quite differently. Because we're giving a shot and then we're basically suppressing the virus, and then we're waiting for it to go low. And then we want the virus to begin to rebound to get this so-called vaccinal effect. I'm going to ask Anne about this in a second, right? This idea that as the virus rebounds with the antibodies around, you get immune complexes, which can turn on the immune system. And in fact, in our clinical trial that Michael directed, the AMPHAR study, we saw that. We saw at the end of the antibody tail simmering of the virus, which we think is good because it induces the immune response. But Anne, in your monkey study, I was wondering if you saw any evidence of that, because I do think your set points, right, when the virus rebounded, it looked to me like they were mainly below 10,000, which is pretty low, isn't it, for
Yeah.
in infants?
Yeah, so we're running T cell assays literally right now. We looked at neutralizing antibody assays to actually see whether we had any or neutralizing antibodies to see whether we had any sort of broadening of the response. And there's two animals. So the infecting virus, this is where we gave the cocktail of bNAbs a week before treatment interruption. There's two and the infecting virus is SIV MAC 251. And there's two animals that had a sort of improvement in their neutralization against MAC 251. And those same two animals also had some evidence of neutralization of SIV MAC 239, which is a tier 3, harder to neutralize virus. Those responses have no correlation with the viral loads, though. So I think that if we do see something with a vaccinal effect, it would be in the T cells, and so we'll see those data very soon. I know in monkeys so far, it's been pretty controversial whether that can actually be detected by the methods that we're all using. So I think the jury's still out there.
I agree with that. Thank you.
Any other questions? All right. Well, thanks so much to Anne and Jesse for really terrific talks. We got great discussion, and thanks again for joining us today, and have a great rest of the week.
Okay, thanks for having me.
Thank
Bye.
you so much for
Okay,
coming.
bye.
Thanks, Anne.